In this lecture, I'm going to be talking about the physiological, environmental, and genetic effects on wood properties. So we're going to talk about a few of the wood properties that we've mentioned earlier, specifically wood density or specific gravity, microfibril angle, and lignin composition and content. I want to point out that for the most part, most of the literature on wood quality and especially effects of the environment and genetics are primarily in conifers. And that's because conifers are really commercially important and for the most part are the type of trees that are used for structural or dimensional lumber, where we are really concerned about traits like specific gravity or microfibril angle or lignin that affect either how they're processed or the strength properties of the product that comes out of the raw material. So most of what I'll talk about is from conifers, but it's probably reasonable to assume that a lot of this variation occurs in angiosperms as well, although the role of tracheids is less important. In angiosperms, a lot of the variation in wood properties probably has to do with size and distribution of vessels and lignification and other properties of fibers. So most of this will focus on conifers. So I want to talk about, we're going to start talking about wood density or specific gravity. Those are different traits, but are relatively interchangeable. Specific gravity is just the density of wood relative to water. So wood that had the same density as water would have a specific gravity of 1. At any rate, determinants of specific gravity in conifers has to do primarily with the structure of tracheids. And variation in specific gravity in early wood, variation in specific gravity in late wood, and then the ratio of early wood to late wood. And those things in turn are for the most part influenced by the cell wall thickness of tracheids in those different zones. Most of the variation in, for instance, specific gravity of a piece of wood or within a ring has to do with the specific gravity of the late wood and how much of that late wood is there. And that's because the greatest cell wall thickness is in the late wood. And so the amount and thickness of cell walls in that late wood really influences those properties. So just to elaborate, this is a nice schematic that shows, for instance, a tracheid in the early wood and a tracheid in late wood in a conifer. So as you can see, the cell wall is thicker in the late wood and thinner in the early wood. Bigger lumen in early wood, smaller lumen in late wood. And you can see that in this micrograph cross section. This is in a pine, red pine, early wood tracheids here, late wood tracheids here. And again, when macroscopically, like in my background picture, the variation between that early wood and late wood is what gives us the visible growth rings. And that's because in the late wood, those cell walls are much more apparent and so reflect light differently. And that also shows you how this late wood would be much more dense than this early wood, just because there's a lot more air in this early wood than there is in this late wood. So the amount of mass per unit volume is greater in late wood. So at any rate, those are the proximal traits that really influence a lot of the variation in specific gravity or density. So this is some data from Larson et al. This is a nice review paper by Philip Larson. He did a lot of work on wood formation in conifers. So there's a lot of data, a lot of papers from the 60s and 70s and into the 80s that Larson wrote on wood formation. So that's some good stuff. At any rate, this shows specific gravity, basically samples of wood that have high specific gravity and low specific gravity. And basically you can see the differences in cell wall thickness in those two wood samples. So the high specific gravity wood sample has somewhat higher early wood cell wall thickness, but much higher late wood cell wall thickness. So that just goes to show you the importance of that cell wall thickness for determining specific gravity. So what triggers late wood formation? So we talked earlier about this relationship between when height growth stops and when the transition to late wood starts. And we can see that late wood transition starts about the time that height growth is slowing. So maybe 5 to 10 days before height growth stops is when late wood formation starts. So that formation, that transition from early wood to late wood happens about the time height growth is slowing. Larson thought that this had a lot to do with reduced soil moisture at about the time height growth was slowing. And while that certainly is partially the case, it's probably not entirely the case. I'm sure there are also hormonal and genetic effects which influence the timing of transition to late wood. And like most physiological properties or responses, it's probably a combination of those internal physiological factors like hormonal balance between growth hormones produced in the apical meristem, along with environmental factors like soil moisture that trigger those responses. Now, certainly irrigation tends to prolong wood formation and the date of late wood cessation later into the growing season. So I think I showed you some data earlier in the lecture that showed that irrigation increased late wood formation and therefore increased specific gravity. So certainly there's strong experimental evidence that moisture content alters late wood cessation, which is when diameter growth stops. So some complicated relationships there. So there's a lot of variation within the stem in most of these wood properties. So variation in the stem in specific gravity, and we can think of variation both radially. So from, if we're, for instance, pull a core at breast height out of a tree and analyze the different rings for wood properties within that core, we will see variation. And one way that I want you to think about that is that when you look at, for instance, the wood near the pith of the stem, you're in the juvenile wood. So these rings early or near the pith are taken from a time when the crown of the tree was nearby. And remember, we talked about how hormonal effects of photosynthesizing leaves plus the apical meristem influences wood formation and influences the juvenile wood core. So a lot of what we're seeing, for instance, in this relationship, when you see that as you go outward from the pith towards outer rings, you see an increase in specific gravity. A lot of that has to do with this developmental response that you see here. As the crown is receding, we're starting to see the formation of mature wood, for instance, at breast height. And if you recall, mature wood has a greater ratio of late wood to early wood. So juvenile wood tends to have less late wood and therefore has lower specific gravity. But as the crown recedes, the mature wood that's formed has therefore higher specific gravity because it has more late wood, and probably also has greater early wood and late wood specific gravity as well. Another thing, and I'm not going to show you any data, but you would see similar responses if you sampled up in the tree. So if you sampled the current growth ring going from the base of the tree to the top, you would see a reduction in specific gravity because you're moving up into the crown. And again, you're seeing the influence of the crown on those outer rings. So if you sampled wood from up here, it would have a lower specific gravity due to the influence of leaves and the apical meristem. Another factor that influences wood properties is degree or intensity of management. When I speak of intensity of forest management, I'm usually referring to silvicultural treatments like fertilization, control of competing vegetation, and so on. And those treatments are applied usually to enhance or speed up the rate of growth of trees. And of course, those types of treatments also then have the possibility of altering wood properties. And the research does show that silvicultural intensity does tend to alter wood properties. So in southern pines, intensive management like fertilization and weed control tends to, on average, decrease specific gravity of early wood and late wood and increase the size of the juvenile wood core. So there's some somewhat negative effects on wood properties. Now, from an economic standpoint, in general, when we're thinking about growing wood, there aren't premiums placed on wood quality. If you're selling a stand, for instance, on the open market to be cut, they won't sample the specific gravity and say, oh, I'll pay you another $1 a ton because these trees have greater specific gravity. So in general, intensive management is still applied regardless of slightly negative effects on wood quality, because again, the effects are not huge and there's not a premium for that better wood quality. So some of these intensive management effects that we see here. So for instance, this shows late wood specific gravity for different treatments. So what we see is that, for instance, the irrigation treatment, which essentially has no nutrients added, or the control treatment have the highest specific gravity versus fertilization or fertilization plus irrigation. Now recall, irrigation, its main effect is to extend the length of the growing season and therefore increases the late wood amount of late wood produced. So that's why that irrigation treatment is pretty high. That and you can see that. I'll show you a graph of percent late wood. So you see that irrigation is pretty high in that. So in general, intensive management like fertilization, especially, and weed control isn't shown here, but we see similar effects. It tends to basically increase the size of tracheids, and therefore they have a larger lumen and maybe a thinner cell wall relative to the lumen size, just because of the more rapid growth under those conditions. So that's the biological effect, but like I said, there also are economic effects that determine whether people choose to apply intensive management or not. There's also genetic effects on wood properties. So for instance, this table shows effects on a number of traits: whole core specific gravity, early wood and late wood specific gravity, and percent late wood across a number of regions. So what you see is that in general, the South Atlantic region has, for instance, higher specific gravity and higher percent late wood than the other more northern regions. Often, excuse me, when you see geographic effects like this is in loblolly pine, which has a very large range. When you see geographic effects, that often means there also are genetic effects. But in this was just plantations sampled in different parts of the geographic range of loblolly pine. So geography and, for instance, seed source was probably confounded in this study. In order to, so we can't say to what extent the environment in the South Atlantic is affecting these properties, which it most certainly is, versus the genetics that are growing in that particular place as well. Those are mixed up together. In order to definitively determine whether there's a genetic effect, you have to do a common garden study. So remember, a common garden study is when we take seed that has adapted to particular locations in different parts of a species range and plant them together in one place. That's a common garden study. So for instance, this is a common garden study in radiata pine. And what you can see is that the seed sources, excuse me, and that's these families, when grown in one location, there's a lot of variation in wood density in those families. So this is, in terms of wood density, quite a large range of variation in wood density. So this is just a frequency distribution of the families, and this shows families with high wood density, families with low wood density. And you can see they're not varying much in size in this common garden, but there's big variation in wood density. So that just goes to show you that, for instance, within radiata pine, there's a lot of genetic variation in wood density. So what that tells us is that we can likely, for instance, design selection programs to select for desirable wood properties like specific gravity in these types of species if we so wish. We would have to do studies to determine whether those traits are heritable, and we'll talk about tree breeding later in the semester, but the existence of this kind of genetic variation is a clue that you might be able to do selection for that desirable trait. Another important wood property trait is microfibril angle. So recall, microfibril angle is the angle of microfibrils relative to vertical in the S2 layer of the cell wall. We call S2 layers the thickest secondary cell wall layer, and therefore determines wood properties of wood cut from that tree. And that more vertical or smaller microfibril angles result in stiffer wood. So it's an important trait in terms of wood quality, because smaller microfibril angles result in stiffer wood, which is an important strength property. So just like specific gravity, microfibril angle varies within the wood. So this is the graph of microfibril angle, so larger to smaller, versus, again, this is radial transect, probably at breast height, from the center out towards the outside of the stem. So you can see both in a fast-growing tree and a slower-growing tree, microfibril angle becomes more vertical as you're moving from the center towards the outside of the tree. So just like the case with specific gravity, this is due to the effects of the crown. If the crown, and again, this goes back to juvenile wood. Remember when we talked about juvenile wood properties, juvenile wood tends to have larger microfibril angles. And that's due to the influence of hormones in the crown of the tree, probably primarily produced by photosynthesizing leaves and the apical meristem, so growth hormones. And those influence the microfibril angles in tracheids formed while they're within the crown of the tree. So these early phases where microfibril angles are less vertical result in, are due to being in the juvenile wood core, whereas these more vertical microfibril angles occur in mature wood after the crown has receded. And again, the same thing would happen if you just took samples from the outer ring going vertically in the tree. As you went up in the tree, the microfibril angle would become less vertical, would become larger, again, because you're getting closer to the influence of the crown. I just wanted to make a side note. This graph is from, I agricultural handbook called Utilization of the Southern Pines by Koch. This is a 1972 publication. You can probably still find it online as a PDF. It is just filled with great illustrations, some beautiful line drawings, and a ton of data about southern pines. Wood quality mainly, it's focused on wood, but there's a lot of information about the biology of wood formation and so on. So a really neat resource to check out. So that's spatial variation. There's also can be genetic variation in microfibril angle. So for instance, this is some data from Roth et al. In a number of different families of loblolly pine. And what you can see is that this is stiffness, a measure of stiffness. But stiffness is related to microfibril angle, remember. So there was significant variation among families in stiffness. So that shows that there's genetic variation in microfibril angle. Just as a side note, this stiffness measure was acquired with an instrument like this. And the way this works is you drive this device, basically a spike, into the tree, and then drive another spike into the tree that has a receiver further up. And then you whack the lower spike with a hammer. And this device measures how long it takes that sound wave to propagate, or that shock wave to propagate within the stem in between the two spikes. And the speed of propagation is related to stiffness. So it's an indirect measure of microfibril angle. That's why this is V squared, that's velocity squared. That is directly related to thickness. So it's a nice non-destructive measure of some pretty important strength characteristic of trees in situ, in living trees, without having to destructively sample them. So another property of wood that's an important, especially for human use, is lignin. Lignin is, as we mentioned before, a complex polymer that acts as glue in tree stems and in other tissues. But lignin is very hard to break down. So this is a picture of the Rayonier Specialty Fibers Mill in Fernandina Beach in Florida. This is a neat place for a tree-oriented person to vacation. It's a beautiful island. It's Amelia Island. And the town of Fernandina Beach is located there. There actually are 2 pulp mills on this little island. They smell a little bit, but if you're a forestry type like me, it's pretty cool to have 2 pulp mills, within walking distance. So at any rate, the Rainier Specialty Fiber Mill is there. And one of the reasons those pulp mills use so much energy and sometimes they're stinky is because they need to use processes that are energy intensive to break down lignin and remove it from the tracheids and fibers in wood, because those fibers are then used in wood products. So at any rate, there are different types of lignin, and 2 of the main types of lignin are S and G lignin. So syringol and guaiacyl lignin. So S lignin is found in angiosperms and is not found in conifers. And S lignin is easier to pulp. It's easier to break down because of its chemical properties. Whereas G lignin is the main type of lignin found in conifers. Conifers don't have S lignin in them. So conifers have lignin that's harder to break down. Angiosperms have both S and G lignin. And because G lignin is harder to pulp, the S to G ratio, so the ratio of those 2 types of lignin in, for instance, angiosperm wood, ends up being an important trait for how easily those woods can be processed in mills. And there's actually genetic variation in the composition of S and G lignin in trees. So here's an example in eucalyptus. So eucalyptus, here's an example of data in angiosperm. So most of the data we've looked at is conifers. Here's some nice angiosperm data showing geographic variation in S-lignin, or actually in S to G ratio, and its variation relative to the mean. You can see there's strong geographic variation in the SG ratio, which tells us that there's actually probably strong genetic variation in the SG ratio. And people that grow eucalyptus for fiber actually have selected for SG ratio. So there are clones of eucalyptus that are used that have been selected to have a favorable SG ratio for pulping. So that genetic variation in lignin type, and in some cases lignin concentration, can be really important commercially. It's natural genetic variation, and I may not be real clear what the selection pressures were that drove that, but it ends up being an important commercial trait that does have genetic variation. This S-lignin, G-lignin thing is so important that there actually are studies in conifers. This is an example where they're looking at actually using genetic engineering to basically create lines of pine that have the ability to make S-lignin, to alter that S to G ratio to make them easier to pulp. There's nothing like this being used, but that's the purpose of this type of study, looking at the genes that control production of S-lignin and then inserting them into pine genome to see, can we make pines that make S-lignin? So a commercial product or genotype would be far down the road, but that's an example of work along those lines.